Okay, hello. Today is the first lecture about biotechnology. So in this first lecture of biotechnology, we'll understand a general idea about biotechnology. What is the science behind biotechnology? What is a major benefit of studying biotechnology, okay? So for the topics of biotechnology, we have the first topic is we'll understand what is biotechnology and what are the challenges of the 21st century, okay? Then we'll go to, this is actually the topics of the whole course. So we'll discuss what is biotechnology workforce. Then we'll talk about gene and genomes, types of mutations, causes and consequences. Then we'll deal with a very important technology, which is recombinant DNA technology, and how that is related to genomics. Then we'll talk about the techniques and the application of recombinant DNA technology. Then we'll talk about genomics and its relation to bioinformatics, and I believe you are taking a course of bioinformatics, and both of them are hot discipline of biotechnology. As well, we'll talk a very good topic on proteins as a biotechnology product, like insulin, for instance. Then we'll talk about proteomics and biotechnology. Then we'll deal with the DNA fingerprinting, one of the major techniques used in forensic analysis. Then we'll understand how we can use the biotechnology in the aquatic system and how we can generate a better yield of fish, for example, for human as a consumable for humans. And as well, we'll talk about the medical application of aquatic biotechnology. We'll talk more details in medical biotechnology. And then we finalize the biotechnology course with the human genome, a project, and we'll understand how they managed and succeeded to understand the human genome. And actually, these are the topics of the course of the biotechnology. So for the assessment, so we'll have on week 7 a midterm for 30% of the mark. And then we have from week 11 to 14, not all, of course, the 3 weeks. It had to be one of these weeks. We'll have like presentations and team-based learning for 30%. And of course, we'll have the final exam, which will be 40%. For the reference of the biotechnology course, actually, it's a very good book, which is the international edition of the Introduction to Biotechnology. And it's a very good book dealing with biotechnology. From this book, you will have 3 chapters: the microbial biotechnology, and the plant biotechnology, and the animal biotechnology. And this actually your homework for the presentation, what will use reference for that for the presentation, which have around 30% of the total mark. For the presentation, you must stick with this template. So the template starts with the first slide, which must contain the university logo, the title of the talk, the course name and code, and submit it to, of course, my name and your name, the student name, and the ID. For the second slide, you will state the objective of your part of the presentation. So I mean the topics you're going to talk in your presentation. Then for the other slide, you need to stick with this format of text. So you have a heading that will be around font 24 Arial bold. The whole body text of the slides will be font 20 Arial. The line spacing single, and the paragraph spacing is 6 points. Before last slide, you will do a conclusion slide of what you have said. And then in the last slide, you'll put the reference, and of course, it will be the name of the book which I just said, the Pearson one, the international edition. So let's talk about the biotechnology century and its workforce. So we'll deal with 4 subtopics in this major topic. We'll talk about what is biotechnology and what does it mean to you, the types of biotechnology, the biological challenges of the 21st century, and the biotechnology workforce. So let's see this image, for instance. You can imagine this is the tip of a pin, the booth, and you can see that it have on this tip around 8 blastomere stage. So the miracle cell or the human embryo, after 3 days of fertilization, you can see it on the tip of a pin. It's a very minute and it's still a miracle cells. These miracle cells can develop to the whole embryo. In addition, there is a lot of stem cells within these embryos that have the potential for treating and curing a lot of human diseases, but by using biotechnology. So scientists are trying to extract these stem cells because these stem cells have the potential to differentiate to every single type of our body cells. And they try to extract these stem cells and to do differentiate it into various types of our body cells. This will help a lot in the process of transplantation. So instead of taking a liver from a donor, you can get some stem cells from your own body and differentiate it into a liver cell and then transplant it into the same body. In this scenario, there will be no rejections, okay, from the body. So for biotechnology, actually, it is using the living organisms or products of the living organisms for human, for the benefit to make a product and to solve a problem. That's biotechnology. The historical example, what already people were doing without knowing that is actually a biotechnology, is the process of fermentation, the process of selective breeding, the use of antibiotics. You can see here how the maize have been developed using a biotechnology from these species that gives you very low yield maize corn, and to develop a very good species of maize that can be eaten or used in the industry. And this, of course, by the help of biotechnology. You can see here, this kind of fish, it's a normal zebrafish. And of course, it got its name, zebrafish, from the look, okay? And this zebrafish, they managed to do another species which actually transparent, totally transparent. So they termed it, or they named it, Casper zebrafish. So by the process of biotechnology, they managed to do this species. So what is the help of this Casper zebrafish? Actually, this Casper considered to be a model organisms, and you can inject this Casper zebrafish with any type of cells, especially you can inject it with the cancer cells, and you look from outside to see where these cancer cells can go, where these cancer cells can migrate inside the body of the fish and infect or metastasize or spread inside the organ. So via the process of selective breeding, you can make zebrafish mutant that lacked the reflective pigment. So normally fish have a reflective pigment on their outer body surface. That's why it gives the looks of the fish. So when you mate two kinds of fish, one of them is lacking this reflective pigment with the normal zebrafish, and over a multiple generation of selective breeding, you will result in a species of a fish that it is lacking this pigment and it looks transparent. And this Casper is transparent. So this feature allows scientists to inject a fluorescent cancer cells. And what is fluorescent cancer cells? Actually, it is cancer cells that you can see it because they emit fluorescence. So you can know where is these cancer cells inside the body of the fish. So when we inject these fluorescent cancer cells into the Casper abdominal cavity, they can track the migration of those cells to specific location in the body. So in this scenario, they can understand where the cancer cells will prefer to go once inside the body. So what biotechnology have, and what is the major trunks that form the big tree of the biotechnology tree? So you need to understand that different disciplines contributes to biotechnology. And any scientist want to work with biotechnology, he needs to understand a bit of every of these trunks. So actually the basic sciences and what you are taking in the college, these are called basic sciences. These basic sciences represent the roots to understand the biotechnology. So these roots are represented by basic sciences. You can see chemistry, physiology, mathematics, molecular and cell biology. You can see a lot of basic sciences that all give the strengths for the understanding of the science behind the biotechnology. Then if we go further, you can see that the trunk for the most biotechnological application, actually, it is majorly the genetic engineering. So you need to understand very well how to do genetic engineering in order to do biotechnology, in order to understand more in the biotechnology. So understanding genetic engineering through, of course, the molecular biology and the analysis of the bioinformatics will lead you to the discipline that you like in future, the discipline that you want to work in future that is related to biotechnology. What about branches? Actually, you can see different branches. It depends on the type of organism you're going to work. It depends on the technology that will be used, and as well the applications. And all of them are actually stems that comes from the genetic engineering basis. So you can see someone will work with environmental and aquatic system, others will work in agriculture biotechnology, other will work in detection and forensic sciences, other will work in drug development, and so on. So from this tree, you can understand what is the roots, what is the trunk that support the understanding of biotechnology in different applications. So what is the more modern examples of biotechnology? And of course, we'll deal with this in details in the coming lectures. If you need to know biotechnology, you need to understand gene cloning. You need to understand genetic engineering. You need to understand one of the major technologies, which is the recombinant DNA technology. And you need as well to understand what is the Human Genome Project. So the examples of application of biotechnology, you can develop a plant that is disease resistant. You can develop food crops that produce higher yield. You can develop, which already developed, it's a golden rice. So golden rice, it's already developed and it's been sold in the market. and it gives more nutrition, and it's a bit expensive than the normal rice. And you can Google it for golden rice. You can see it looks golden and have a lot of nutrient values. You need to understand how genetically engineered bacteria can degrade environmental pollutants. So that's a way of bioremediation. So you can remove any pollutant in the environment by using bacteria through understanding the biotechnology. And these are the applications. If we look closely here, you can see how they manage to map the chromosomes. That's example chromosome 13 and chromosome 21. You can see that the scientists managed to understand which region of the chromosomes will affect which type of disease. Like this region will affect breast cancer, other regions will affect pancreatic cancers, and so on and so on. So this is like a map for chromosome 13 and chromosome 21. And of course, both of these figures are for knowledge. So the Human Genome Project led to the identification of nearly all human genes, and as well they mapped their location on each chromosome. And you can follow, if you are more interested, you can follow this link to know which gene is associated, which disease is associated with which chromosome. So let's see here the number of the biotech drugs that scientists managed to do and their relation to the diseases. You can find that cancer actually have a lot of produced biotech drug. So what do we mean with biotech drugs? So biotech drugs, we mean that drugs that is developed, that they are developed by the use of the living organisms. So it's not purely chemical, because as we said initially, biotechnology is a science which deals with the using of living organisms or product of the living organism. So when we talk about biotech drug, so that this is a different class of drugs that been developed for these diseases. So you can see that cancer actually acquires of most around like 500 different drugs, followed by AIDS, followed by infectious disease, and so on. Okay, so there is a booming in the types of the biotech drugs for the different disease. And you can see that this disease has many more drug candidates than hepatitis. So if you look to hepatitis, you can see that hepatitis have like around 48 drugs, while cancer have around 500 drugs. So why cancers get a lot of number of biotech drugs over, for example, hepatitis C? Because this cancer, because hepatitis C affects only less number of people worldwide than the cancer. So it's more demanding to have more biotech drugs for cancer over the hepatitis C. And as well, cancer can affect many different organs, while hepatitis C only affects the liver. And that's a very logic and obvious why cancer have more drugs than the hepatitis C. The same story goes with diabetes, for instance, because diabetes as well have low number of drugs, because we can say that it's only affecting the pancreas, for instance, or the blood sugar level, not like cancer, which can affect a lot of different organs inside our body. So let's hear here an introduction about how they generate some of the biotech products. So you can see that you can use genetically modified cultured cells to make up any protein of interest, any protein of interest. You initially clone, of course, we'll talk later in the lectures how they do this process in details. So they clone the gene of interest in something like a circle, which is called a vector, and they introduce this vector with the gene of interest. They insert it inside a bacteria or mammalian cells. And they start to grow these bacteria or mammalian cells in the fermenter, a large fermenter, and they induce the expression of the protein of that gene of interest. And these cells, either mammalian or eukaryotic or prokaryotic, mammalian or bacteria, start to grow and divide and express the protein which is inserted by the gene of interest, the gene of your interest. And then this protein, once expressed, you can isolate this protein. You can extract this protein. In these cases, we can't call it protein. We must call it recombinant protein. Recombinant protein. Why recombinant? Because it has been made artificially, and the gene of interest have been inserted into a vector or plasmid. So that's why this structure is called recombinant plasmid. So the protein which is formed is called recombinant protein. In this case, this recombinant protein can be isolated, and there's a lot of techniques that you can easily isolate this recombinant protein. And this is a perfect example of what actually happening for insulin. So they do the insulin with the same process. So you can see here, this is the insulin, and they do the process to have recombinant insulin, and now insulin have been sold a lot cheaper than before, because of the process have been easier, and the process have been less cheaper, because initially they were getting the insulin from the sheep or horses, which was very expensive to be sold. But now, since they express the insulin in the bacteria and they have huge fermenters, so this lowers the cost of insulin and increase the yield of insulin for the human use. So we can see here, this is the top. This is the top biotechnology drugs with sales above 1 billion. So actually these drugs now already been sold and have been developed by the understanding and the use of modern technology. So you can see here some of the famous companies like Roche, like Johnson & Johnson, like Sanofi, and these are all companies that they can develop all these drugs, and it is used for all these diseases. Okay, so as well, this table is for knowledge. So you can see here as well how some of the examples of the product of the modern modern biotechnology. So these are actually proteins that have been developed by using the gene cloning and the recombinant technology, and hence it's named now recombinant protein. So you can see they can manage to form to do the insulin. So you can see here. So you can see insulin, one of the products or the recombinant protein that used to treat diabetes. They manage as well to do blood factor 7, which is a clotting factor used to treat hemophilia. They manage as well to develop one of very important recombinant protein, which is the monoclonal antibodies. And these monoclonal antibodies is used now for treatment of variety of diseases, including cancer and inflammation or autoimmune disease like arthritis. So actually, these are examples of the recombinant protein have been developed and the applications of these recombinant protein.